Now that spliceosome that was discussed in the previous mini lecture, the one that was there for the processing out of the introns and the ligating together the exons, that spliceosome is special. Why is it special, you ask? Well, let me tell you. That spliceosome was composed primarily of RNA, short little RNA strands. The RNA strands, I say that, and a lot of you will picture in your minds figures from the textbook where they show the RNA strand as a nice straight line. And in reality, it's far from that. Short RNA strands, even the long ones, will start bending around and complementary base pairing with itself, and they'll form these little loops, these straight parts, and it'll loop around and all sorts of weird three-dimensional shapes. So a straight line of RNA can loop around and form a three-dimensional shape. And I have told you from the first lecture on in this course, shape dictates function. So these RNAs can take on a three-dimensional shape that gives them a function. Turns out that spliceosome is actually what we refer to as a ribozyme. A ribozyme is an enzyme that doesn't meet the criteria we've talked about in all the previous chapters. Whenever I've said an enzyme in previous chapters, it's always been in relation to a polypeptide or a protein that has a specific shape so it can do a specific function. Ribozymes are composed primarily of RNA molecules that have taken on a three-dimensional shape. Shape dictates function. They now have a function. Any amino acids, any polypeptides present is just there to hold the different RNAs together. The protein or anything protein-like is there just for holding the overall structure together. The actual work is done by RNA molecules. What's interesting is one gene, one polypeptide. One gene, one mRNA, one polypeptide, according to our central dogma. Well, not always. There is a thing referred to as alternative splicing. What happens when the spliceosome will loop out an intron, but instead of joining exon A to exon B, because it got rid of the intron between them, it joined exon A to exon C, a different one. B gets looped out also. They refer to this as alternative splicing. So you can get different mRNAs from one gene. Turns out the exons are generally going to be the blueprint, the description on how to make what is referred to as a domain, a specific part of a polypeptide. Think of it as a domain gives the instructions on how the secondary structure will fold to make an alpha helix. The next exon calls for how to make a beta sheet. The next exon, a beta sheet. The next exon, a loop. And it just goes on and on and on like this. So the different portions of the overall polypeptide they refer to as domains. Each domain comes from a different exon. Exon 1, 2, 3 with introns in between. Each exon calls for a different section within the overall peptide. Due to signaling, due to alterations, signals come down through, one of these signal pathways due to a response triggered by a receptor being bound will tell the spliceosomes to do alternative splicing. So instead of polypeptides 1, 2, 3, what if it's only 1 and 2? 3 gets lost. What if it's only 1 and 3? 2 gets looped out. One gene, multiple mRNAs that could be made that look different, which will result in different polypeptides.